Welcome back to Biochemistry Learning on the Go.
Back again.
For Podcast 2D, we're doing a deep dive into
Oh.
polysaccharides,
I like it.
specifically their structure and function and clinical significance
You know,
as biochemistry professors.
this is right up our alley.
These are near and dear to our hearts.
Absolutely. Polysaccharides,
Yeah.
they're those complex carbohydrates that I think sometimes get a little overshadowed by their simpler cousins, the monosaccharides and disaccharides.
Yeah.
But trust me,
No.
they're just as fascinating, if not more so.
Absolutely. I always tell my students, don't underestimate the power of a well-placed glycosidic bond.
Absolutely.
Today we'll be exploring how those bonds and the intricate structures they create translate into a mind-boggling array of functions,
Yeah.
from energy storage to cellular communication.
And we'll even touch on how those functions can go awry,
Right.
leading to various clinical conditions.
Exactly.
So buckle up because we're about to embark on a whirlwind tour of the polysaccharide world.
Yes, so first let's define what we're dealing with.
Okay.
We're talking about carbohydrates with 10 or more monosaccharide units linked together.
Right. And just like with any good taxonomy, we can break them down into two main categories.
Okay.
You've got your homopolysaccharides,
Mm-hmm.
made up of the same repeating sugar unit,
Okay.
and then your heteropolysaccharides, where you have a mix of different sugar units.
So kind of like building with Legos.
Exactly.
You could have a tower made entirely of the same type of brick, or you can get fancy and mix things up to create more complex structures.
And just like those Lego creations, the type and arrangement of the building blocks.
Right.
In this case, the sugar units determine the overall properties and function of the polysaccharide.
Okay, so let's start with the homopolysaccharides,
Okay.
the ones built with a single type of sugar brick.
Right.
First up, we have starch,
Starch.
the plant kingdom's go-to energy storage molecule.
Starch is like a plant's pantry,
Yes.
packed with glucose ready to be used when photosynthesis isn't churning out enough energy.
I love that analogy.
Now, starch itself is made up of two main components,
Okay.
amylose and amylopectin.
Ah, yes, the dynamic duo of plant energy storage. I remember when I first learned about them, I was struck by how a simple difference in branching could have such a profound impact on their properties.
Yeah, and it all boils down to those glycosidic bonds.
Okay.
Amylose is a straight chain of glucose units linked by alpha 1/4 glycosidic bonds.
Okay.
Think of it as a long, unbranched necklace of glucose.
While amylopectin takes it a step further, adding alpha-1 milli-6 glycosidic bonds that create branches along the chain.
Right, more like a bush with multiple stems and branches compared to amylose's single linear strand.
Exactly.
And that difference in branching has significant implications for how plants store and utilize energy.
Yes.
Amylose being tightly packed is excellent for long-term storage.
Makes sense.
Right.
That-
Whereas amylopectin, with its branch structure, provides more surface area for enzymes to break down the glucose when the plant needs a quick energy boost.
So it's like having multiple access points to that glucose pantry?
Exactly. Now, if we shift our focus to the animal kingdom,
Okay.
we encounter glycogen,
Yeah.
our own version of starch, but with even more branching.
Glycogen is like the supercharged high-octane fuel reserve for animals,
I like it.
primarily stored in the liver and muscles.
Yes.
It's ready to be mobilized at a moment's notice for those fight-or-flight responses or bursts of intense activity.
Imagine a densely branched tree with countless twigs.
Right.
That's glycogen. The highly branched structure allows for rapid enzymatic breakdown, releasing glucose into the bloodstream when we need it most.
Okay, so we've got plants storing energy as starch and animals storing it as glycogen.
Yep.
Both are built from glucose, but differ in their branching patterns and how quickly that energy can be accessed.
Precisely. Now let's talk about cellulose.
Okay?
Another homopolysaccharide built from glucose, but with a completely different function.
Cellulose, the tough fiber stuff that gives plants their structural integrity.
Yep.
I always find it fascinating that it's made of the same building block as starch and glycogen, yet it's completely indigestible to us humans.
It all comes down to the type of glycosidic bond.
Okay.
Cellulose is composed of glucose units linked by beta-1,1,4 glycosidic bonds. Unlike the alpha-1,4 bonds in starch and glycogen, our digestive enzymes can't break those beta bonds.
Which is why cellulose passes through our digestive system, largely intact,
Hmm.
acting as dietary fiber.
Exactly. And while we can't extract energy from it like we do from starch fiber, plays a crucial role in our gut health, regulating bowel movements and even helping to prevent certain diseases.
Speaking of indigestible polysaccharides, let's talk about inulin.
Okay.
This one always intrigued me. A chain of fructose units found in plants like chicory onions and garlic.
Inulin is fascinating because while we can't digest it, it serves as a prebiotic,
Right.
nourishing the beneficial bacteria in our gut.
It's like a food source specifically for those good gut microbes, helping them thrive and contribute to our overall health.
Precisely. And inulin has another interesting application.
Huh?
It's used medically to test kidney function.
Really? How does that work? I'm always curious about the unexpected applications of these molecules.
Well, because inulin is not metabolized by the body, doctors can inject it into the bloodstream and then measure how much of it is filtered out by the kidneys into the urine.
So it's like a tracer molecule, allowing doctors to assess how efficiently the kidneys are filtering waste products through the blood.
Exactly. It's a clever way to get a snapshot of kidney function without relying on more invasive procedures.
Okay, so we've covered starch, glycogen, cellulose, and inulin, all homopolysaccharides, with diverse functions, despite being built from just one type of sugar unit.
Right.
Before we move on to the more complex world of heteropolysaccharides, there's one more homopolysaccharide I wanted to touch on, dextrin.
Dextrin.
Yeah.
It's often overlooked, but it plays a significant role in various industrial and food applications.
Okay.
Essentially, dextrin is what you get when starch is partially broken down.
So it's like starch in a transitional state, caught between its original form and its complete breakdown into glucose units.
Exactly. And those partially broken down chains of glucose give dextrin some unique properties.
Okay.
It's slightly reducing, meaning it can donate electrons in chemical reactions.
Interesting.
And it's not fermentable by yeast, unlike its parent molecule starch.
Interesting. So it seems like even these simple homopolysaccharides have a lot going on.
Absolutely.
I'm already starting to appreciate the incredible diversity of structures and functions that could arise from just a handful of sugar building blocks.
But hold on tight.
Okay.
Because when we delve into the realm of heteropolysaccharides,
Right.
those with a mix of different sugar units,
Yes.
things get even more intriguing.
Okay, I'm ready to dive into that world of heteropolysaccharides.
All right.
But first, let's take a moment to let those homopolysaccharides sink in.
Good idea.
We'll be back in a flash to explore the even more complex side of the polysaccharide family.
Sounds good. Welcome back to our deep dive into the world of polysaccharides.
Back again.
I hope you've had a moment to digest all those fascinating homopolysaccharides we discussed earlier.
It's amazing how much variety we see, even when using just one type of sugar building block.
Right. But now, let's shift gears and explore the heteropolysaccharides.
Okay.
These molecules are composed of different sugar units,
Right.
resulting in an even wider array of structures and functions.
So if homopolysaccharides are like building a tower with a single type of Lego brick, heteropolysaccharides are like using all the different shapes and colors to create something truly unique.
That's a fantastic analogy.
Right.
And one of the most important groups of heteropolysaccharides is the glycosaminoglycans, or GAGs for short.
It's
You might remember them from your medical school days.
yes, GAGs. They're essential components of the extracellular matrix, that intricate network of molecules surrounding and supporting our cells.
Precisely. And their unique structures give them remarkable properties,
Okay.
allowing them to play diverse roles from shock absorption to cellular signaling.
Okay, I'm intrigued.
Mm.
What makes GAGs structurally distinct from the homopolysaccharides we discussed earlier?
Well, unlike starch or cellulose, which are built from repeating glucose units, GAGs are made up of repeating disaccharide units.
Disaccharide units. So two sugar molecules link together, and these pairs then repeat to form the long GAG chains.
Exactly. And these disaccharide units are special because they typically contain an amino sugar and a uronic acid.
Now, remember those terms from biochemistry, but let's refresh our listeners' memories.
All right.
What exactly are amino sugars and uronic acids?
An amino sugar is essentially a sugar where one of the hydroxyl groups, those OPOX-OH groups you see in sugar molecules, has been replaced with an amino group NASH-NH2.
So it's like taking a regular sugar and adding a bit of nitrogen to the mix.
Precisely. And a uronic acid is a sugar that has been oxidized at the terminal carbon, forming a carboxylic acid group, NASH-COH.
Okay, so we're taking these modified sugars, linking them together as disaccharide pairs, and then repeating those pairs to form long chains.
Right.
That's how we get those unique GAG structures.
Exactly. And these modifications, particularly the addition of those carboxylic acid groups, give GAGs a strong negative charge.
Which I imagine has a significant impact on their interactions with other molecules in the extracellular matrix.
Absolutely. Those negative charges attract water molecules, creating a hydrated gel-like environment.
Okay.
This is crucial for many of their functions, from providing cushioning in joints to acting as molecular sieves.
Okay, so we have these long negatively charged GAG chains floating around in the extracellular matrix.
Yes.
What exactly are they doing out there?
Well, they're incredibly versatile.
Okay,
One of their primary functions is to provide structural support and maintain tissue integrity.
right.
For example, hyaluronic acid, a very well-known GAG,
Yeah, hyaluronic acid.
is a major component of cartilage, providing resilience and shock absorption.
Hyaluronic acid, it seems to be everywhere these days, from skincare products to joint injections.
It's a true multitasker.
Yeah,
Its ability to bind water molecules makes it incredibly effective at keeping tissues hydrated and resilient. Think of it as a molecular sponge soaking up water and creating that cushioning effect.
so hyaluronic acid is like the shock absorber of our joints, keeping things running smoothly.
Right.
What other important GAGs should we know about?
Another one you've likely heard of, especially in the context of osteoarthritis, is chondroitin sulfate.
Okay.
It's another crucial component of cartilage, providing structural support and helping to resist compression.
So chondroitin sulfate is working alongside hyaluronic acid to keep our joints healthy and functional.
Exactly. And then there's keratan sulfate, which plays a vital role in maintaining the transparency of the cornea in our eyes.
Uh-huh.
Imagine if our corneas weren't clear, we wouldn't be able to see.
That's a great example of how the specific structure of a GAG dictates its function.
Hmm.
We've got hyaluronic acid keeping our joints hydrated, chondroitin sulfate providing strength and resistance, and keratin sulfate ensuring we can see the world clearly.
And there are others, each with its own specialized role. For instance, dermatan sulfate helps maintain the shape of the eye, and heparin sulfate is involved in cell signaling and growth, acting as a kind of communication hub on the cell surface.
Wow. It's amazing how much diversity we see within this one group of heteropolysaccharides.
It really highlights the elegance and complexity of biological systems. Now, there's one more GAG I want to mention, and it's one you've definitely encountered in your medical training.
Okay.
Heparin.
Heparin, the famous blood thinner.
Hmm.
I remember learning about its crucial role in preventing blood clots.
Exactly. Its unique structure allows it to bind to and activate antithrombin III, a protein that inhibits key enzymes involved in the coagulation cascade.
So it's not just about providing structural support and hydration.
Yeah.
GAGs are also involved in regulating crucial physiological processes like blood clotting.
Absolutely. And this diversity of function makes them incredibly important from a clinical perspective.
Right.
Defects in GAG synthesis or breakdown can lead to a wide range of disorders, from skeletal abnormalities to blood clotting disorders to even vision problems.
It's clear that understanding GAGs is essential for anyone working in the medical field.
Absolutely. But before we move on, there's another fascinating aspect of GAGs that I want to highlight. While some GAGs, like hyaluronic acid, exist as free chains, most GAGs are actually attached to proteins forming proteoglycans.
Proteoglycans. They sound familiar, but I need a refresher.
Yeah.
What are they, and why is this attachment to proteins so significant?
Imagine a bottle brush. The core protein is like the handle, and the JAG chains are the bristles sticking out in all directions.
Okay.
This unique structure gives proteoglycans remarkable properties.
So it's like taking those long, negatively charged GAG chains and attaching them to a protein scaffold.
Right.
What does that achieve?
Well, for one, it creates a much larger, more intricate structure. Think of it as taking a bunch of those bottle brushes and linking them together to create a giant space-filling network in the extracellular matrix.
So these proteoglycans are not just individual molecules, they're assembling into larger complexes that contribute to the overall architecture of the extracellular matrix.
Exactly. And because those GAG bristles are so good at attracting and holding water, proteoglycans form gel-like matrices providing cushioning and support for tissues.
So they're like the molecular scaffolding and shock absorbers of our tissues all rolled into one.
That's a great way to put it.
Right.
And those proteoglycans can even interact with each other, as well as with other components of the extracellular matrix, to form even larger, more complex aggregates.
It sounds like a real molecular dance party going on out there in the extracellular matrix.
It is. And this intricate network of molecules is essential for maintaining tissue integrity, regulating cell behavior, and mediating a whole host of physiological processes.
This is all so fascinating, but before we get too carried away with the wonders of GAGs and proteoglycans, I want to make sure we touch on another important group of heteropolysaccharides.
Okay.
The glycoproteins.
Ah, yes, glycoproteins.
Yeah.
They often get lumped together with proteoglycans because both involve carbohydrates attached to proteins. However, there are key differences.
Right. I remember the glycoproteins have shorter carbohydrate chains compared to the long repeating JAG chains found in proteoglycans.
Exactly. Glycoproteins typically have short-branched oligosaccharide chains attached to specific amino acid residues on the protein.
So instead of those long, bristle-like gag chains, we have shorter, more intricate carbohydrate structures decorating the protein surface.
Precisely, and don't let their smaller size fool you. Those oligosaccharide chains are incredibly diverse and play essential roles in a wide range of biological processes.
Okay, I'm ready to delve into the world of glycoproteins. But first, let's give our listeners a moment to absorb all that incredible information about GAGs and proteoglycans.
Well,
We'll be back shortly to explore the fascinating functions of those sugar-coated proteins.
all right, sounds good.
Welcome back to our polysaccharide exploration. We've covered so much ground, from the energy powerhouses of starch and glycogen to the structural marvels of cellulose and the intricate world of glycosaminoglycans.
It's been quite a journey through the diverse landscape of these complex carbohydrates, and we've arrived at our final destination, glycoproteins. These fascinating molecules, while not strictly polysaccharides in the classic sense, showcase the incredible versatility of carbohydrates in biological systems.
Right, we were just discussing how glycoproteins differ from proteoglycans, those bottle brush-like structures with long gag chains. Glycoproteins have shorter branched oligosaccharide chains attached to their protein backbones.
Exactly. Think of these oligosaccharides as intricate sugar decorations adorning the protein surface. And these decorations are far from superficial. They play crucial roles in a wide range of cellular processes.
Okay, I'm all ears. What kind of roles are we talking about?
Well, one of the most prominent functions of glycoproteins is in cell signaling and communication. Think of those oligosaccharide chains as molecular flags or ID badges, allowing cells to recognize and interact with each other.
So it's like a sophisticated language of sugars allowing cells to communicate and coordinate their activities.
Precisely. For example, many hormones are glycoproteins, and those sugar chains are essential for binding to their specific receptors on target cells.
It's like a key fitting into a lock. The shape and composition of the oligosaccharide chain determine which receptor it can bind to ensuring that the hormonal signal is delivered to the right place.
Exactly. And it's not just hormones. Many cell surface receptors, enzymes, and even antibodies are glycoproteins relying on those sugar chains for proper function.
Speaking of antibodies, those immune system workhorses are also glycoproteins, right?
Absolutely. And those oligosaccharide chains play a critical role in determining the specificity and effectiveness of antibodies.
So those sugar chains are helping antibodies recognize and bind to specific antigens, those foreign invaders that trigger an immune response. It's amazing how these tiny sugar structures can have such a profound impact on our immune system's ability to fight off infections.
It really highlights the elegance and specificity of biological systems, and it's not just about recognizing and targeting foreign invaders. Glycoproteins are also involved in regulating cell adhesion migration and even development.
So they're influencing how cells interact with their environment, move around, and even differentiate into specialized cell types.
Precisely. Think about the development of an embryo. Cells need to communicate, migrate, and organize themselves to form complex tissues and organs. Glycoproteins are essential players in orchestrating these intricate developmental processes.
It's mind-boggling to think that these sugar-coated proteins are involved in so many fundamental aspects of life, from cellular communication to immune defense to embryonic development.
It's a testament to the incredible versatility of carbohydrates and their ability to fine-tune the function of proteins. And from a clinical perspective, understanding glycoproteins is crucial for diagnosing and treating a wide range of diseases.
Right. We were discussing earlier how defects in GAG synthesis or breakdown can lead to various disorders. I imagine the same holds true for glycoproteins, given their involvement in so many crucial processes.
Absolutely. For example, certain types of cancer are associated with altered glycosylation patterns on cell surface proteins. These changes can influence tumor growth, metastasis, and even drug resistance.
So by analyzing those glycosylation patterns, doctors could potentially diagnose cancer earlier or even develop more targeted therapies.
Exactly. And there's growing interest in developing drugs that target specific glycosylation pathways or even using engineered glycoproteins for therapeutic purposes. It's a rapidly evolving field with enormous potential.
It's fascinating to see how our understanding of these complex carbohydrates is paving the way for new diagnostic and therapeutic approaches.
Indeed. And it all starts with appreciating the fundamental role of carbohydrates, not just as energy sources, but as essential players in virtually every aspect of cellular function.
Well, on that note, I think we've reached the end of our deep dive into the world of polysaccharides. It's been an incredible journey exploring the diverse structures, functions, and clinical significance of these often overlooked molecules.
I couldn't agree more. It's been a pleasure sharing our passion for these fascinating carbohydrates with our listeners.
And to leave you with some food for thought, consider this. How might the emerging field of glycomics, the study of all carbohydrates in an organism, revolutionize our understanding of health and disease? It's a question worth pondering as we continue to unravel the intricate world of these essential biomolecules. Thanks for joining us on this deep dive into the world of polysaccharides. Until next time.
Keep exploring the fascinating world of biochemistry.